right. Slideshow. Okay. So this is the second video in the Bio EOC crash course. This section we are going to be talking about heredity, which is basically all of your genetics, DNA, RNA, pedigrees, all of those lovely things. So with heredity, here are grade level expectations that we're going to be focusing on, mainly biotechnology, pedigrees, DNA, RNA, genes, chromosomes, all that jazz. All right. So first things first, biotechnology. If you should have your review session 2 heredity packet in front of you, and go ahead and copy down these notes. So biotechnology is technology that uses biology. Shocking. So some examples are cloning, stem cell research, genetic testing, genetic engineering, GMOs, which is changing the genetics of crops like corn or rice to make them better for production. So some pros: it will increase production and make money for people who use it. Another pro is that it could cure disease. There are also some cons with biotechnology. Number one being, is it ethical? So if you ever get to a question on the EOC that's talking about biotechnology, your first thought should be, is this an ethical thing that's going on in this question? Next one, is it fair and equal for everyone? And is it safe? Is it going to mess up something with whoever's using it? All right. Once again, you can pause this and take the notes down if you are not writing as fast as I'm talking. All right, number one, scientists have genetically engineered monkey DNA, but have not genetically engineered human DNA. The best explanation for this situation is that what? A, the technology is genetically engineer humans has not been explored. B, human reproduction is very different from that of other mammals. We know that's not true because it's sexual reproduction. C, there are many ethical problems involved in genetically engineering humans. Or D, genetically engineering humans would take too long. So right off the bat, it's talking about genetic engineering. I'm going to zoom in on the answer choice that is asking if it's right or wrong. And it is going to be C. There are a lot of problems involved with people thinking that it's not right to genetically change the genes of humans. All right, 2. A current direction in medical research involves analyzing the DNA of patients to determine the best courses of treatment for their diseases. In the near future, which part of human society would most likely be affected first by this technology? So what this is asking is, if there's a big breakthrough in science, like a big, huge cure in cancer, who are going to be the first people that are benefiting from this cure? The answer, more oftentimes than not, is going to be people who can afford it. Wealthy people are going to be the first to benefit from this technology, which is definitely not right, but that is the current situation. So, which answer choice basically sums that up? A, most of society, because DNA analysis is such a powerful technology. No, that's not true. Not everybody's going to get this treatment first. B, people in industrialized countries, so like the United States, because these procedures will be expensive at first. C, all people worldwide. We know right off the bat, not all people are going to get this at first. Or D, primarily the third world, which is the poorer countries, where there's a greater incidence of disease. Now, D is the people who should be getting this, but that is unfortunately not how the world works. And unfortunately, the people who are going to benefit from this first are people in industrialized countries. All right. So on the DNA, on the biotechnology sheet, there is another question on there that you can go ahead and do on your own. But I'm going to keep moving on to DNA, RNA, protein synthesis. All right. So go ahead, copy down these notes. We have DNA. The function is the genetic information. Basically makes you who you are. I also gave you a couple other notes in there with the structure and strands of it and the bases. We know that DNA has A, which pairs with T, and G, which pairs with C. And we remember it because D stands for double strand. It doesn't really stand for that, but that's how I remember it. RNA, we have 2 types of RNA. Go ahead and write both of those down. We have our mRNA, which carries the message from the DNA to the ribosomes. And we have our tRNA, which brings amino acids to the ribosomes. RNA is only one-stranded, and if we look at our bases, the only base we do not have in RNA is T. We instead replace it with U. So what that looks like, and go ahead, I'm going to have you draw this in the blank space below the chart of DNA and RNA. So first things first, we have our DNA to mRNA. This process is called transcription, and it occurs in the nucleus. So I drew my nucleus as a circle. This is the first thing that has to happen. The reason for this is because mRNA is the only one that can leave the nucleus and take that message. DNA is stuck inside and cannot leave. So next thing that happens is our mRNA is going to be changed into amino acids. This occurs in translation, and it occurs in our ribosome. So I drew my ribosome as a square. So what that looks like in a problem, say we have this strand. This is going to be our DNA. We know that because it has a T in it. And our first step is we have to get it to mRNA. So our T is going to pair with an A. Our A is going to pair with a U. C's are all going to pair with a G. So now we have our strand of mRNA, and it can piece out of the nucleus. It can leave. It's going to go all the way over here to our ribosome and split into these groups of 3 called codons. We are then, and you have the chart in your packet. It's on page 4. And I'm going to explain how to do this in a second. But we are then going to switch from our mRNA to our amino acids. So this one happens to become MET-GLY. What that looks like, go ahead, turn to page 4 in your packet right now, and you'll see this chart on page 4. So what this looks like, we're going to model this by going through the problem to the bottom left of the chart. So we have our first strand, AGU. We are going to always look for our first letter on the left side, our second letter on the top, and our third letter on the right side. So what that looks like, first letter is A. We find it over here where our first base is, A. Second letter is G. We find that up here. Your first step is to narrow down which square you're going to find your amino acid in. So A and G, where they meet, is in this lovely square. We know our amino acid is going to be in this square. The way we determine which one it is by looking at that third letter and following it across. So U, our amino acid is going to be SER. Please rewind this if you are confused, or call me over to explain it a little bit more. This just takes practice, but once you figure out how to do it, you're going to be fine. So if you go through all of this, you go through the next 3. These are the amino acids you should end up getting. CGA, we have C over here, G over here, so we know it's going to be in this top square. And then A, we followed across, it's ARG. All right, so at this time, you have some sample problems in the whole DNA, RNA, protein synthesis section of your packet. Go ahead and do those on your own, and make sure you turn them into Ms. McGrath for some feedback on them. Alrighty, I'm going to keep moving. So Punnett squares, this is how we do genetics. So for right now, go ahead and take a second to copy down these lovely definitions. First one we have is genotype. All that is the genetic makeup. It's just the letters. Whenever we're describing genotype, we're using letters. But in real life, you do not look at someone and describe them using letters because that would be ridiculous. So we use what's called our phenotype. Phenotype is the physical appearance, so what it looks like. So instead of saying you look big A, little a, I'm going to say you have brown hair. Ooh, shoot. Homozygous. Homo means the same. Same trait, so it's going to be the same letter. Either 2 big letters or 2 little letters. Hetero is different, so you're going to have 1 big, 1 little.